Hello everyone, welcome to today's live broadcast, Multidimensional High-Throughput Imaging with the new ImageXpress Micro Confocal System. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Molecular Devices. Molecular Devices is one of the world's leading providers of high-performance bioanalytical measurement systems, software, and consumables for advancing protein and cell biology. With over 130,000 placements in laboratories worldwide, their instruments have catalyzed brilliant scientific research described in over 80,000 peer-reviewed publications. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenters. We are proud to welcome Jane Hesley and Grisha Chandy. Jane Hesley is an applications scientist for cellular imaging at Molecular Devices. She has more than 6 years experience developing cell-based applications using ImageXpress high-content imaging systems and MetaXpress analysis software. Most recently, her focus has been on utilizing biologically relevant cell models for toxicity studies. She has a BS in animal science and MS in biology. Grisha Chandy, PhD, is the Senior Product Manager for Cellular Imaging at Molecular Devices. Grisha's scientific background is in cell biology, physiology, and biophysics. He has used and developed imaging techniques and technologies throughout his career. Grisha has been with Molecular Devices for over 10 years, driving launches of several ImageXpress systems and MetaXpress software, and most recently, the launch of the ImageXpress Micro Confocal system. I will now turn it over to Jane.
Thank you, and welcome to our webinar. My name is Jane Hesley, and I'm an imaging application scientist at Molecular Devices. In this webinar, I will cover some confocal applications, specifically 3D spheroid screening and no-wash homogeneous assays. Next, Grisha Chandy, the product manager for imaging, will introduce you to the features of our new ImageXpress Micro Confocal imager. First, in case you're not completely familiar with high-content and high-throughput imaging, I'll spend a minute giving an overview of the workflow and the potential applications. Traditionally, biologists would treat some cells with drugs or growth factors and put them on a microscope slide after staining them or in some way marking their response with fluorescent proteins. Or they may be looking at morphological changes with transmitted light. Now, instead of peering at them through the eyepiece, they'd take pictures or images to study and get a sense of whether or not the cells reacted to the drug. Even if the acquisition with a conventional confocal microscope is automated, the hands-on time can be multiple hours, and the skills to run a successful experiment may be high. Under the question mark are the possibilities of what happens next, because it's not common anymore that the images are simply judged by the researcher's experienced eye. By using a high-throughput imaging system, experiments can be just as easily run on anything from microscope slides to 1536 well plates, with no compromise in image quality. The ImageXpress Micro system can be set up in a few minutes, and the biologist can walk away while images are acquired and even sent immediately for analysis by the integrated software. Once numbers are generated for the parameters of interest, responsive cells or wells can be identified, statistically analyzed, plotted, and conclusions drawn. Since all images and measurements are stored together in a database, the burden of keeping the data organized is removed from the scientist, and the results can be shared within a networked company or organization. The hands-on time is dramatically shortened due to the automated and seamless integration of acquisition, analysis, and viewing of results. Briefly, there are hundreds of applications that can be done with automated imaging. Obviously, these slides list just a subset of them. In these images, you see an example of co-cultured liver and fibroblast cells, a high magnification image that could be used to analyze multiple subcellular structures, and an example of angiogenesis in a gel matrix. This slide shows more examples, including sample images of neurite outgrowth, a 3D spheroid, and transgenic zebrafish. A nice example of small organism imaging is this Drosophila larva. On the left is a video made from time-lapse acquisition at low magnification, and the right illustrates how confocal acquisition of individual Z-planes allows you to completely separate and visualize individual structures within the Drosophila larva's head. Why use 3D models? Cell models for assays are becoming more complex in order to better mimic the in vivo environment and provide greater predictivity for compound efficacy and toxicity screening. Naturally, there's an increasing interest in using three-dimensional assays for modeling cancer and tissue biology. These three-dimensional in vitro models may span the gap between two-dimensional cell cultures and whole animal systems, and can provide unique perspectives on the behavior of stem cells, developing tissues and organs, and tumors. While the overall goal of using 3D models is to improve predictivity, the purpose of this first study was to optimize cell culture and develop new imaging and analysis methods that can be used for compound screening through assessment of multiple phenotypes in human cancer 3D models. Specifically, we optimized the workflow for spheroid formation, staining, and image acquisition for 96 and 3D4-well assay formats for three commonly used cell lines, HCT116, DU145, and HepG2. We have developed multiparameter assays and analysis methods for spheroid cultures that provide subcellular resolution and characterization of single-cell and overall spheroid phenotypes. There are some challenges associated with efficiently capturing the structural detail and identifying marker localization in complex 3D models such as spheroids, whole organisms, cells grown throughout a gel matrix, or in the presence of multiple cell types, which may actually be layered upon each other. I'll focus mainly on the example of using spheroids in a screening assay for part 1 of this presentation. Then in the second part, touch upon some other homogeneous screening assays and explain the benefit of using confocal optics for acquiring the images to obtain the information needed. When cells are grown in an extracellular matrix, such as matrigel, there's generally unavoidable background fluorescence, if not from the gel itself, then from the difficulty of adequately washing out the fluorescent stains used on the cells. Here's an example of two images of some PC12 cells grown throughout about an 80 micron thick layer of matrigel. While acquiring the images with wide field optics still shows nice cellular detail, the background fluorescence from the matrigel is apparent. Imaging the same cells with confocal optics illustrates the benefit of the thin sectioning ability of the confocal acquisition. See how the background fluorescence is eliminated, which will often make analysis easier. I don't want to give you the impression that confocal optics are always necessary for high-content imaging. For many, if not most, flat 2D assays with cells and monolayers, wide-field imaging is the correct option. Even for identifying punctate objects within cells or making morphological measurements, wide-field imaging of a single plane is the simplest and fastest screening mode. All the features I'm going to illustrate for 3D confocal imaging are available for use with wide field, including bright field imaging, time-lapse acquisition, as shown here, sorry it was so fast, and collecting and analyzing stacks of images in the Z-plane. Confocal optics will improve image quality in cases where you need to reject fluorescent backgrounds, such as in no-wash assays, or if the media is contributing fluorescence. It's also great for those thick samples I mentioned in the previous slide, so you can adequately resolve different layers of cells in space. This example shows our software's 3D rendering of images collected from the bottom up through the middle of a spheroid that was stained with live dead markers. The other one shows an example of stepping through the planes of cells grown in matrigel. So now we'll address some of the technical challenges of using cancer spheroids for high-content imaging. The first one will be culturing the spheroid you plan to use, followed by some insights into staining of these structures. Next will be techniques for imaging the stained spheroids and then analyzing those images. You will want to go from reproducibly culturing spheroids to segmentation of the images you acquire to deciphering the data you generate in your assay. There are numerous methods for creating spheroids. I'm using an illustration here from 3D Biomatrix of the gravity or hanging drop method. In the hanging drop method, a plate with tiny holes in the bottom in the configuration of a microplate is placed above a humidified chamber. Cells in media are pipetted into each of the holes, and the drop containing the cells hangs suspended by surface tension. The cells gather in the bottom of the drop and readily aggregate into a spheroid. Spheroids can be imaged right in the drop or after forcing it into a microplate. Some other ways to create spheroids is to grow them on a scaffold, such as the one illustrated in this low magnification image of an array of spheroids on a special plate from Toyo Gosei, where they utilize techniques to repel the cells from the areas between the arrays and attract them to specific areas on the plate. Or by using a 3D gel matrix like Matrigel. Many cell types will spontaneously form spheroids when not confined to a microplate surface. There are some challenges for these methods when it comes to media exchange, compound addition, and possibly requiring transfer of the spheroids to another plate for imaging. We've been successful using ultra-low attachment plates from Corning that allow much easier cell culture, media change, and compound addition. The workflow is similar to the standard culturing of cells, using a little extra care as if these were non-adherent cells. For example, we don't attempt to completely remove media from the wells before adding fresh. Here you can see the movie of spheroid formation from HCT 116 colon cancer cells over a weekend, imaged at 3-hour intervals. Spheroids are formed within 24 hours but are nice and compact and ready to treat after 2 days. This is an example of a 96-well plate of spheroids after 7 days. Note that every well has a single spheroid and the size and shape are quite consistent, with diameters varying less than 10%. The spheroids self-center and can be captured in a single field of view with 10x or 20x magnification. This means the instrument doesn't have to find the spheroid in the well or stitch multiple sites. We have overcome several challenges to make the assay convenient and feasible for automated HCS. One of the challenges with spheroids is the staining steps. First of all, dye penetration, especially of labeled antibodies, may be impaired by tightly packed cells. Secondly, wash steps may either move the spheroid off-center or disrupt its shape, especially if it has been weakened by the compound treatment. So for this viability assay, we used Herxt for a nuclear stain, to stain the nuclei in all the cells, calcine AM to indicate cell viability, it will stain metabolically active cells, and Ethidium homodimer for dead cells. It stains the nuclei of cells with damaged external membranes. We used live cell staining, one mix containing all 3 dyes, and no washing before imaging. We have also successfully used caspase 3,7 for measuring apoptosis, and a couple of different mitotracker dyes to gauge compound effect on mitochondrial integrity. Here are a couple of graphs showing our optimized incubation time and dye concentrations. We decided a 2 to 3 hour incubation of the live spheroids at 37 degrees C gave the best intensity signals. We also generally ended up using about a 2 to 4 times higher concentration of the dyes than we would normally use for our 2D cell toxicity assays. To get a representative idea of what is occurring throughout the spheroid, we set up the ImageXpress Micro Confocal to acquire multiple Z-planes. There's a finite distance a fluorescent microscope can reliably acquire clear images into a thick structure due to the interference of a large number of cell layers. Because of this, we generally image from the bottom of the spheroid to just over halfway through it to get a consistent and reproducible sample stack. The scientist has the choice to either save each and every one of those Z-planes or else to collapse the images into a single 2D image on the fly. You can also analyze either each Z-plane or else just the 2D projection image. Here's an example of what I was just describing. Confocal images of a spheroid stained with Herx were taken at indicated distances from the well bottom. As the image plane moves up from the well bottom toward the spheroid center, regions of distinct nuclei appear first in the center, at the surface of the spheroid in contact with the plate bottom, then forming a ring pattern that increases in diameter until it reaches some maximum associated with the outer surface of the spheroid. The maximum projection image is generated from 11 individual images taken 20 microns apart. Nuclei segmentation is shown for the corresponding 60 micron and 160 micron images, and also for the 2D maximum projection image. The individual nuclei in the masks are given a random false color. This graph plots nuclei counts for a single spheroid measured from the 11 different Z images taken at indicated distances from the plate bottom. It shows that the largest sampled area that can be segmented was found between 60 and 100 microns deep into the spheroid. The in-focus nuclei counted past 100 microns were the ones seen on the spheroid's perimeter. This bottom graph was generated from the nuclei count in the maximum projection image from a single spheroid based on the number of individual images in a Z-stack taken over a 200 micron distance. In other words, if you took 10 Z-planes to cover the 200 micron distance versus 30 Z-planes and built a 2D maximum projection, the nuclei count did not get any better. From these results, we decided to typically image our spheroids using 11 Z-planes 20 microns apart. These two videos are to compare Z-stacks acquired with a 20x objective using either wide field mode or confocal mode. Basically, it is just showing you stepping through the Z-stack from the bottom to the top of the spheroid. In this case, I used an algorithm to generate a best focus 2D projection from those previous stacks of images. As you can see, the confocal images result in lower background fluorescence and therefore more accurate counting of the nuclei. In several experiments, we collected both wide field and confocal stacks of the same spheroids and compared the nuclear counts. On average, the software is able to identify about 80% as many nuclei from a wide field 2D projection as from a confocal 2D projection. Now to show the assay put to use. This is a screenshot of a spheroid plate treated with 6 compounds with increasing concentrations as you go up the plate. This was a live cell assay stained with the mix of 3 dyes as described previously. As you can see, different compounds cause various phenotypic changes in spheroids, from stunted overall size in the staurosporine-treated wells to beginning to disintegrate at the top of the doxorubicin column. You can see several examples here that show the mask of the object and also masks of individual cells scored for the presence of different markers. We easily set up a custom analysis module that would identify and report out only the parameters we wanted to look at. We wanted to consider a number of readouts that characterize morphological changes like spheroid width and area. Also, we evaluated total cell count in the spheroid by Herx stained nuclei and used the viability markers to count the number of live cells, dead cells, and metabolically active cells. While these masks show what remain of the spheroid based on Herx stained nuclei, you may also analyze the cells in the entire field of view so that the dead or apoptotic cells may be accurately counted. This bar graph shows several different measurements for 6 treatments compared to the untreated control, which is shown in bright red. The top graph is illustrating the count of cell-by-cell results, where the bottom graph shows data collected on the spheroid's diameter, area, total area of live cells, and total calcineum intensity within the spheroid. Another way to look at the data is shown by the results from this 96 well plate screen of 4 compounds. The number of live cells or other readouts can be used to define IC50s for each compound. To wrap up this application section, I hope we've shown you some useful information that will help you if you're interested in confocal imaging of spheroids. I think we've shown that 3D spheroids can be reproducibly grown, compound treated, and stained, that high-content confocal imaging of spheroids is suitable for compound screening in microwell plates, that spheroid phenotypes can be characterized using morphological analysis and population counting. And if you want further information, I encourage you to read this paper, High Content Assays for Characterizing the Viability and Morphology of 3D Cancer Spheroid Cultures, which contains a lot of this information and even more. Now I'm going to move along to the homogeneous antibody binding assay, commonly known as FMAT. I'm going to first explain how this assay works because it's not commonly used in all cell-based imaging labs. Either cells that express some kind of target or beads that are coated with a target are floating in suspension in a microwell plate. An analyte is added, and in this case we're using a species-specific antibody called human antibody, as the analyte, as it would be if someone wanted to test the production of a human antibody from a hybridoma colony. Then a fluorescently labeled secondary antibody against the analyte, in this case it's goat anti-human IgG, labeled with an alexafluor, is added to the mix. Often these are incubated for several hours to allow complete binding, and for the cells or beads to settle to the bottom of the plate. It looks here like the fluorescent signal will be concentrated onto the beads, assuming there is some human primary antibody to complete the sandwich. If you image those wells and measure the amount of alexafluor 48 signal associated with the beads, you see a curve generated in a dose-dependent manner, meaning as the concentration of the analyte increases, the intensity on the bead increases. But here's the surprising part for those unfamiliar with this no-wash assay. The signal of the highest analyte concentration is actually decreasing. I'll quickly show you why in the next slide, but I want you to know that when we are evaluating this assay for lower detection limit, we are omitting the points that are to the right of this peak. Now the ratios aren't completely right in this illustration, but it shows you that in the presence of a very high analyte, which is the reddish human primary antibody, all the binding sites on the beads might be filled up with the human antibody, and all the binding sites on the secondary can be filled up with the human antibody as well. This can occur to the extent that there's no sandwich formed between the capture surface, the bead, and the fluorescent detection, the secondary antibody. This results in lower signal on the beads, which shows as the right side of that previous graph. It can also result in a higher background in the media around the beads or cells. With that explained, here are some actual images taken at 10x magnification of 6-micron beads with the fluorescently labeled antibody sandwiched onto them, while there is still an excess of the labeled antibody in the media or buffer, since remember we don't wash any of the antibody off. The wide field image on the left and the confocal image on the right are scaled to the same intensity values, and by drawing a line through the images and plotting the pixel intensity seen along this line, you can see that you get much lower background signal if using confocal optics, as well as a much higher signal to background ratio of the beads versus the background. You might guess that with the improved signal to background ratio, the lower detection limit will be much better if confocal optics are used. We often see 5 to 10 times better sensitivity in an antibody binding assay by using confocal images. We calculate an LDL as the lowest concentration we tested that is 3 standard deviations above the average background bead intensity. That's the bead in the presence of the secondary antibody, but not the primary human antibody. Here we saw 0.3 nanograms per mil with confocal versus 4.7 nanograms per mil with wide field. As you know, these absolute sensitivities will be dependent on each antibody's purity, specificity, affinity, off rate, etc. These homogeneous assays can also be done in cells. This example shows CHO cells expressing an unnamed receptor and combined with a ligand, and then detected with a fluorophore-labeled antibody to the ligand. These were imaged with a 20x Plan-Apo objective, and we see a clear benefit of confocal imaging to segregate cell membrane staining, especially against a high background. In summary, the ImageXpress Micro Confocal system provides background rejection that is necessary to measure homogeneous assays. Confocal imaging delivers a lower detection limit for antibody binding to beads that's more than 5 times lower than the corresponding wide field images. Confocal imaging also enables greater resolution of antibody binding to cell membrane receptors. I concentrated on the applications that make use of the confocal side of our automated imaging system, but the solution is also relevant for your 2D wide-field imaging screens. If you collect exquisitely high-quality images, it makes segmentation and analysis easy using MetaXpress software and either the turnkey or customized application modules. Then you evaluate the measurements that are output and plug them into a AcuityXpress data analysis program for identifying the hits or trends in your experiments. I will leave you with an eye full of pretty images while we transition to Grisha to tell you more about the new ImageXpress Micro Confocal system. Thank you.
Thanks, Jane. That was a great overview of a couple of applications enabled by our new product. As a reminder, I'm Grisha Chandy, the product manager for the high content portfolio at Molecular Devices. I will now give you a short introduction to the new product, the ImageXpress Micro Confocal system. As a reminder, the system is both a confocal and wide field automated imager that enables applications in the screening, research, and live cell space. The movie on the left is taken over 16 hours in a 3D 4-well plate, with each well repeatedly imaged approximately every 20 minutes. You will see some cells, some of those highlighted with the orange circles, going through cell division. Additionally, the marker used goes to the nucleus in a puncture structure when the cells are going through the S1 phase. As I mentioned, we are repeatedly visiting each well of the 3D 4-well plate instead of sitting and imaging in one well. The movie illustrates a few things. One, the system is imaging in confocal mode, and the cells are happy, alive, and going through their normal cell division. Two, the system has the stage precision and accuracy to come back to the same area of the well repeatedly over these 16 hours, enabling the type of long-term live cell assays that our ImageXpress customers routinely do to do these multi-day live cell experiments and track cells over time. We additionally wanted to ensure that you had a confocal system that met the sensitivity and throughput requirements of the screening workflow. The image on the left is taken at 4x magnification of a 3D 4-well. You will see that in one shot you capture the entire well with that single image. With the two-color assay, you can acquire the whole 3D 4-well in 12 minutes. On the right, we have a unique feature on our ImageXpress systems, as well as the new ImageXpress Micro Confocal system, and that is at 4x with a 1536 well plate, we can capture 4 wells in one shot. This allows for really ultra high throughput imaging in the 1536 well format, because though you're capturing 4 times more wells, you don't need to capture 4 times more sites, and you can do that in the same 12 minutes. This will roughly translate into greater than 160,000 wells per day in a 24/7 environment automated system. What do you get with the ImageXpress Micro Confocal system? As I mentioned, you get both a confocal and wide field imager. The system comes configurable with 1 to 100x magnification, extremely large field of view, one of the largest on the markets at 1.96 millimeters squared at 10x. That enables you with one shot of the 10x objective to get an entire 1536 well imaged, and an extremely bright solid-state light engine for the reliability and sensitivity needs of a confocal system that is expected to run 24 hours a day, 7 days a week. Access more complex biologies quickly with a system that's compatible with any clear bottom plate type, 1 well to 1536 wells, round or flat bottom, transverse plates, and slides. Note that much of the spheroid data that Jane presented on the application sections of this webinar used round bottom plates that not all HCS systems can image reliably. We can do that. The system is also capable of greater than 3 log dynamic range. The stage resolution is less than 100 nanometers, and you saw the accuracy and precision that allows us to return to where those cells are dividing. And the system comes with both high-speed laser and image autofocus. Not all systems on the market have both that capability. Laser autofocus helps with the speed and not bleaching your sample, and the image autofocus is when your biology, your cells, are not in a reliable place above the well bottom, and it might vary, and you might need to find that within an image autofocus. The ImageXpress Micro high-content imaging systems are flexible to meet your research needs. They come with a variety of options, including complete environment control, including humidity, gas, and temperature control, true phase contrast, transmitted light, and bright field options, and fluidics for onboard liquid handling. Additionally, the systems can be outfitted with a robotic plate handler for those 24/7 screening requirements. As I mentioned earlier, the system has a number of objective lenses to meet your application needs, going from small animal to subcellular resolution. You can purchase the systems with from 1x to 100x air objectives, 20 to 60x phase objectives, and 40 to 60x oil immersion objectives. And all objectives are user changeable. The system is unique in coming with AgileOptix spinning disk technology that allows you to choose the mode that is right for your research. AgileOptix technology uses a bright solid-state light engine, proprietary spinning disks with advanced optics and optimized pinhole geometries, as well as a high-sensitivity SCMUS detector. Mode 1, wide field imaging, gives you the fastest throughput possible. Mode 2 is for confocal imaging. We offer either a 60 micron pinhole for basic confocal imaging or a 42 micron pinhole for high resolution confocal imaging. And lastly, we offer a high throughput confocal imaging mode, which is a unique 50 micron slit technology that enables you to image with the speed of wide field. So how fast and sensitive is the system in reality? To show you that in this webinar, this is a movie taken real time of the system taking Z-stacks through a Matrigel thick sample in 3 colors, DAPI, FITZ, and Texas Red, with the overlay shown next to it. As you can see, and these exposure times are in 100 to 200 milliseconds, hundreds of milliseconds. As you can see in this confocal mode, you can see these crisp images coming up real time, a number of planes in each color, and being displayed live for you, essentially live for you. And so that gives you an impression of what the throughput would be, how sensitive it's going to be for your applications. So I've told you a little bit about the system, and Jane gave you an overview of 2 applications. What you're seeing pop up here are a variety of other applications that are enabled by the system. The top left here, you'll see some neuronal images that were neurons in tissues. On the right here, you'll see some cells that was a Z-stack through a Matrigel. These are nuclear puncta. These are hepatocytes grown in a multiple cell culture system. This is a brain or neural system of a zebrafish embryo, and here is a homogeneous antibody binding to the membrane of a cell membrane of a cell. And many of these applications can be sped up to be done at greater than 40,000 wells a day at high resolution in confocal mode. The ImageXpress systems are also controlled by MetaXpress software, which is an integrated image acquisition analysis package. And with MetaXpress 6 and 6.1, which just came out, which control both the ImageXpress Micro XLS and the ImageXpress Micro Confocal systems, you get an integrated package for your acquisition analysis needs. Built into the MetaXpress software acquisition analysis interface is the ability to acquire and analyze Z stacks. So here you're seeing the acquisition window for a multi-well plate, and you're able to set the bottom where your focus is, the top of your Z acquisition. You can vary the number of step sizes and the number of steps that will do through that Z, depending on the size of your spheroid or your matrix gel. And here's a zoom of that window. Additionally, you'll be able to select whether you only collect the 2D projection or the Z series and 2D projection, and how you are going to project that image. There are 4 different ways to project that Z image. And that is all part of the package you get when you get MetaXpress 6 and 6.1 with one of your ImageXpress Micro systems. At the heart of MetaXpress software is the image analysis toolbox that enables a variety of analysis routines to address the continuum of from turnkey to flexible routines that customers need. So on the left, we're showing an example application module for those routine assays, like maybe cell cycle or counting nuclei or neuroid outgrowth. On the right, we're showing our most powerful journals or macros that allow a wide variety of flexibility from controlling the hardware and making intelligent decisions based on analysis routines. And in the middle, we have our custom modules will give you the flexibility of journals, but the turnkey, once one is created, the turnkey nature of application modules, because once an application module is generated in a custom module, those can be shared with others and reused with very little intervention. So on the application module, canned routines, I'm showing 6 examples: cell cycle, neuroid outgrowth, all the way to cell viability. Those are the raw images, and these are the segmentation. And there's 19 application modules available that are these canned routines. But you can also build your own application modules, whether it's looking for mitotic spindles and centrosomes, doing label-free cell proliferation assays, not just finding neurites, but finding puncta that are near neurites, and doing migration assays. Our solution is a complete imaging workflow when you purchase that from molecular devices, starting with the ImageXpress systems for the acquisition, the MetaXpress for system control and setting up your analysis routines, MetaXpress Power for the ultra high throughput analysis that you might have need for in a screening environment, AcuityXpress for data mining, all tied together with our data management solution, MTC Store. So there's no absolute requirement for importing and exporting data from our solution, but you can bring third-party data into the system as well as automate the export of that data from our solution. So we're bringing to market an automated system for all your imaging needs. It's fast, reliable, flexible, got spectacular image quality that you'd expect from a confocal system. It's got the sensitivity you need, complete workflow solution from image acquisition to data mining, and we've based it on the ImageXpress Micro with more than 1,000 publications out already on the market. And this system just builds on the ImageXpress Micro portfolio. So on that note, we'd like to get your thoughts and take questions, and thank you for participating in our webinar.
excellent presentations. Thank you for bringing that information to us. During the question and answer session, a series of poll questions will appear on your screen. Please take a moment to participate. At this time, we would like to open up the Q&A. Grisha had to step out, and we would like to introduce Deepat Roxakoltai, who together with Jane Hesley will answer any questions you have. Deepat is the application scientist at Molecular Devices, and he has been supporting the high-content imaging product portfolio for over 5 years. Before we get started with all your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question is, you mentioned confocal could benefit a homogeneous apoptosis assay. What do you mean?
Okay, I'll take that. This is Jane. So for homogeneous assays, using confocal optics allows you to reject the background. And often in apoptosis assays, whether it's in 2D cell culture or in 3D, you'll have a lot of cells that are floating away or lifting off the bottom. And by being able to do Z-stacks and reject the background, it allows you to get a better count of the cells that have been marked with the apoptotic marker, as well as any other dead cells that are not really attached to the cell bottom, the well bottom.
What other types of cells have you tried to form spheroids?
Okay, and I'll take that one also. We haven't done an exhaustive list, but pretty much every cell we've put into the bottom of these low attachment round bottom plates have formed spheroids. Sometimes a little bit of, what do I want to say, some protein will help them to form spheroids, but I would say in addition to the cells I already talked about, we've done U2OS cells, and we've had success with induced pluripotent stem cell-derived cardiomyocytes.
How long can you keep cells alive for live cell assays?
All right, we, in-house, I showed you a video of growing spheroids over a weekend, so that was about 2 days and 8 hours, I think. And then we've also had customers who've successfully done their assays in ImageXpress Micro Confocal for up to 7 days.
Can you describe some of the automation options available?
I'm going to let Deepat answer that one.
Molecular Devices offers several different robotic arms and collaborates with several different vendors that we have for robotic integration. Your best solution will probably be to contact your local sales representative and application scientist for any additional information. Basically, any systems that are currently integrated or have been integrated with ImageXpress Micro previous versions, the XLS, the XL, or the standard, will also be compatible with the ImageXpress Micro Confocal. Thanks.
About the objectives, is 100 times oil immersion available?
I'll answer that one as well too. So the platform is compatible with a wide range of Nikon objectives, and we do offer some oil immersion objectives. All of the 100x is available as well, 60 and 100. Thanks.
Is the MetaXpress software compatible with any other platform?
We are able to, the software is able to import and analyze any type of TIFF format or image, most image formats. We also have a couple of solutions for being able to integrate the image formats from different vendors, such as from different microscope platforms into MetaXpress in an automated way through our MDC store database, if that's desired.
And about how long does it take to complete a confocal Z-stack of a single spheroid using 1120 steps?
I'm sorry, Judy, will you repeat that question?
Sure. About how long does it take to complete a confocal Z-stack on a single spheroid using 1120 steps?
Okay. That would depend on the exposure time you need for the dye that you're trying to measure, but generally stepping through maybe a second or so or less for one spheroid for a per well for a wavelength. For example, the nuclear stains I was showing.
Great. Could the ImageXpress be sterilized, for example, with peroxide?
We do have some customers that have, put our systems into greater than BSL1 or BSL2, and the decontamination procedures are pretty standard. I don't know about peroxide in particular, but we've, typically used bleach or a, 70% ethanol iso, mixture, which was more common for us. Sorry.
Great. What dye do you use to show S-phase cells in division?
Okay, I don't think in my experiments I didn't specifically show S-phase, although if you're talking about the video that was shown in Grisha's where he was showing cell division and the live cell assay, those were some special cells expressing chromobodies in the nucleus. And I can't say that I know a lot about them, but I'm sure you can look them up on the web, chromobodies.
Can you do these assays on fixed spheroids?
Yes, there's no reason you cannot. I'll give you the caveat that we're doing most of our work by hand, and it's pretty difficult to fix the spheroids and have them stay in the center of the bottom of the well. But yes, there's no reason you can't, if the dyes are fixable, that you could image the fixed spheroids.
And bear with me, I think I asked you this one earlier, but let's just go again. Is the MetaXpress software compatible with any other platform?
Yes, it is. It's available to open any type of TIFF format or many other commonly used image formats used by other microscope vendors.
Okay, we'll pause for just a moment here to see if we have time for another question or two. Okay, here we go. How do you remove the background from the picture in order to quantify the fluorescence?
All right, so you don't necessarily have to remove the background as long as what you're trying to measure is higher than the background. And our software algorithms in MetaXpress do a really great job of picking up signal that is, very with a low threshold of intensity. And then, of course, if you collect your images with confocal, the background anyway will be reduced, any background fluorescence that's above and below the plane that you're collecting.
And more questions are coming in, so just give us a moment here to thread them through the system. How long does it take to analyze a 384 well plate?
So the time it takes to analyze a plate will depend on the complexity of the analysis desired, but we're also able to, so it depends on if you're doing a simple count nuclei or if you're doing if you're analyzing a multiplexed assay. So it definitely can can vary. We do have an option for implementing parallel processing and multi-threading of analysis, which can speed up your analysis to keep up with the acquisition. So, for example, if your plate took 15 to 20 minutes to acquire, you could also analyze that plate within the same time frame by implementing multi-threading.
Will you be showing this system at the SLAS meeting in, will you be showing the system at the SLAS meeting in January or the HCA meeting in February? And will you have user group meetings at these conferences?
We'll definitely be showing the instrument at those two meetings. We will not have a user group meeting at SLAS, and I'm not positive about HCA. I'm sorry, you'll have to watch our website, I suppose.
Is 40x and 60x oil as in the slides, or 60x and 100x oil as just mentioned?
In most of the slides I showed you, the PowerPoint slides, I guess you're talking about, I used air objectives in all of those, in 40x and my 60x images if I had some, and then which is what's compatible with high throughput. And if you want to use oil objectives, you certainly can, and like we said before, we do sell some of those objectives.
Okay. So we'll pause for just a moment. Questions, or perhaps we will wrap. Just give us a second here to see if we have time to take another question or two. I think we will stop here. I would like to once again thank Jane, Vipat, and Grisha for their presentations. Do you have any final comments for us? Vipat and Jane?
No, I guess if there are any more questions that we didn't get to, I see a couple that we've been giving some thought to. We can answer these people individually. There's no problem with that.
And Vipat, did you have any closing thoughts?
No, we're just really excited about the new platform and hoping that everyone has a chance to ask and get the information that they would need out of it to see how it would help with their projects. And if they have any further questions, your local sales rep, any of your application scientists, or you can direct them again through the marketing team through our website for any inquiries as well too. Also, just for current users, we have a lot of, for current users who are interested, you can contact us, and then there's we have a lot of resources available on our website, for finding out more information, such as our user forum as well for our software users.
Great. And I know we've started to wrap up, but can I just throw one more question at you? And it is, what do you do if the fluorescence of the background is high and the fluorescence of the signal is low?
So this is something typically that we would approach this question either for if you're a current user, our tech support or our application scientists would work with you on. There's only so much that you can do with acquiring the there's only so much that you can do with using the instrument, and we might try to suggest or work with you on going back to the experimental procedure and seeing if there's anything that might be optimized there. I'm sure that most of our users have done a lot of assay development, but there might be some tricks or tips that we've done in the past to be able to boost that. Additionally, outside of the experimental and what we can do on the hardware side, there's also maybe some things. There are a lot of tools in MetaXpress that we could use to be able to pull out some of that signal, even if it is low. So there are a lot of tools available in MetaXpress to be able to do that. One good example would be our homogeneous assays. So if you would go to our website and look up either our posters or our application notes, there might be some tips, even if you're not doing those exact assays. We might be able to provide some tips. And then if this is a question that you need further information on, please feel free to contact us directly, and maybe we might be able to help. Thank you.
Well, thank you. Thanks again to our speakers, as well as our sponsor, Molecular Devices, for making it possible to bring this presentation to you. I'd also like to thank the audience for joining us today and for their interesting questions. And as Jane said, questions we did not have time for today will be addressed by the speakers via the contact information you provided at the time of registration. Today's webcast will be available for on-demand viewing through April 2016. You will receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.